Hello students, welcome to EPG Pathshala. I am Sangeeta Negi from Department of Biotechnology, MNNIT, Allahabad. Today we are going to talk on module Introduction of Carbohydrates from paper Structure and Function of Biomolecules 1. Well, in today's lecture we will study what are the carbohydrates and their classification, and we will do in-depth study of their functions and underlying biochemistry. Further, we will explore the significance and application of carbohydrates. And following objectives we have set to learn depth study of the carbohydrates. First, what are carbohydrates? Classification of carbohydrates and functions of carbohydrates, underlying biochemistry of carbohydrates, and significance and application of carbohydrates. Well, students, this is all about carbohydrate in short. Well, carbohydrates are composed of carbon, that is shown in the black beads, hydrogen and oxygen. They are basically hydrates of carbon and may be classified into three groups: mono, di and polysaccharides. Polysaccharides may be homopolysaccharides or hetero-polysaccharides. In homopolysaccharides, it is composed of single kind of monosaccharides, whereas in heteropolysaccharides, it is composed of different kind of monosaccharides. For example, of homopolysaccharides, cellulose, starch and glycogen. And in heteropolysaccharides present in various different forms like glycosaminoglycans, mucilases, vegetable gums, pectins, etc. They may be present, sugar may be present in its derivative form and play important role in functioning of the cells. They are present in plants, fruits and various milk products and in bacteria or fungus, basically in all kind of living beings. They play important role in cell functioning like cell-cell interactions, cell-cell recognition. They play important role in building blocks and as a structural stability like hemicellulose, pectin and cellulose, and they are a good source of energy like glycogen, starch for future. It can be stored for a long time. Well, as we know, carbohydrates are simplest biomolecule in the universe and they are most abundant also. And they are present in 3/4 part of the biomass. So basically in chemical structure, they are composed of carbon, hydrogen and oxygen in a ratio of 1:2:1, and molecular structure formula is CS2O and 2N, and on the combustion they yield carbon dioxide and water. Basically, they are hydrates of carbon comprised of multiple hydroxyl groups with one aldehyde or ketone as functional group. Naturally, they are present in sugars, starches, gums and cellulases and amino sugars, etc. Carbohydrates can be classified in a broad way into three groups. First is monosaccharide, second di and oligosaccharides and polysaccharides. Monosaccharides are further classified on the basis of the carbon numbers present in that, like trioses, tetroses, pentoses, hexoses, heptoses. And oligo and disaccharides are based on the composition, like maltose, sucrose and lactose are having two units, means they are disaccharides, and maltotrioses, raffinoses, glycans are oligosaccharides. And in polysaccharides, starch, glycogen, cellulose, chitins are placed because they are made up of many sugars, more than 10 sugars basically, and they make a polymeric chain may be branched or unbranched. Well, students, let's describe about monosaccharides. That is the first group, and they are very composed of simple carbohydrates, means they cannot hydrolyze further, and they are the basic unit of polysaccharides. Their empirical formula is CS2O multiplied by N. Minimum value of N is 3, known as trioses, where N equal to 4, 5, 6, 7 are called tetroses, pentoses, hexoses and heptoses respectively. Monosaccharides are classified as aldoses and ketoses, containing an aldehyde and ketone groups respectively. In aldoses, suffix oses and in ketoses, suffix eulose is used as an indicator. A 4-carbon ketose can be termed as tetrulose. Few most common and abundant monosaccharides are ribose, xylose, glucose, mannose, fructose, tagatose, galactose, etc. Monosaccharides are commonly present in all fruits and plants naturally. And disaccharides. Disaccharides contains two sugars joined by an O-glycosidic bond. Three commonly found disaccharides are sucrose, maltose and lactose. Sucrose is common table sugar and composed of glucose and fructose, which we take daily in tea and in foods. Both maltose and cellular are composed of two glucose molecules linked by different bonds. And lactose, present in milk, is composed of galactose and glucose. Well, in second group, oligosaccharides are also kept. Carbohydrates which can hydrolyze into few monosaccharides are oligosaccharides. Generally, they are composed of 3 to 9 simple sugars. And trisaccharides, maltotriose, is composed of 3 glucose molecules. Example of 3, 4, 5 sugar oligomeric polymers are raffinose, stachyose, verbiscose respectively. They play a very important role in cell-cell recognition and signal transduction and many more. Oligosaccharides like glycans are important components of plasma membrane of animals and play important role in cell-to-cell recognition, as I already told you, and also play important role in regulation of transportation of foreign substances through cell membrane. They are found naturally, generally in small amount in many plants. Plants with large amounts of oligosaccharides are chicory roots, from which most commercial inulin is extracted. Onions, garlic, legumes, asparagus, jicama, leeks, wheat and other plant foods. Well, oligosaccharides are classified on the basis of number of simple sugar present as follows: maltotriose, maltotetrose, maltopentulose, linear dextrins, limit dextrins, fructans or fructooligosaccharides. So maltotriose means they contain 3 sugar molecules linked with alpha-1,4 glycosidic linkase, whereas in maltotetrulose 4 sugar molecules are linked with alpha-1,4 glycosidic linkase. In maltopentulose 5 molecules of sugars are linked with an alpha-1,4 glycosidic linkase. In linear dextrins, hexasaccharides with 6 or more glucose molecules linked with alpha-1,4 glycosidic linkase are present. In limit dextrin, which consists of small branched sugar chains linked with alpha-1,6 glycosidic linkase. The fructans are linear polymers of fructose and most abundant storage form of carbohydrate in plants next to starch and sucrose. They are synthesized and generally stored in vacuoles, which accumulates in fruits, stems, bulbs, and tubers of plants. Well, fructans are useful in promoting growth of microflora in guts. In general, they promote growth of bifidobacteria, which produces natural antibiotic against E. coli and streptococcus. We know E. coli and streptococcus are main cause of deadly diseases of stomach. That's why our parents advises us to take fruits in daily diet. Here we can see the fructose molecules are linked with each other by glycosidic or glycosidic linkage, and they may be present in up to 3 to 9 molecules. Well, student, other oligosaccharides are galacto-oligosaccharides, which are known as GOS. It is short chain of galactose molecules and occur naturally but are less digestible to humans. And Menon oligosaccharides, which is also known as MOS, are oligomer of mannose molecules and are present in cell walls of Saccharomyces cerevisiae. MOS are good source for animal feed to improve gastrointestinal health, energy level and performance. Many other derivative oligosaccharides are also present like derivatized sugars like amino sugars, glycolipids, glycoproteins, etc. They play many functional roles in animals. As monosaccharides and polysaccharides playing very important role in cell functioning. So it needs a diversification. Therefore, many sugars get derivatized into different forms to give different kind of functions to the cells. So one is very important, that is amino sugars such as D-glucosamine and D-manosamine and D-galactosamines are derivatized oligosaccharides in which OH group is replaced by an amino group. And best example is chitin, which serves as hard shell exoskeleton in many arthropods. We can see all beetles, crabs, crustaceans, lobsters, crabs, shrimps and other jellyfish, shellfish, all are having very hard shell which provide them protection and prevent from their prey. So oligosaccharides attached with the protein and lipid which are known as glycoprotein and glycolipids to give functionality to the cell wall or cell membrane. They are used as a marker and by many organisms for cell recognition. As when oligosaccharides are attached with the protein or lipid of plasma membrane, they are protruding outside to catch the signal, and they pass that catch signal across the plasma membrane to the inside cascade so that the signal can be processed for further. For an example, blood, which have 4 types of specificity A, B, AB and O due to the presence and absence of glycoproteins. Well, students, let's talk about polysaccharides. Generally, polysaccharides are long chain of monosaccharides. Either they may be a straight chain or may be a branched, which are linked, means the monomers of the polysaccharide are linked with each other by glycosidic bond. The solubility of polymers depends on the number of branching present in it. When it is highly branched, that means it will be more soluble. For example, glycogen is much soluble than the starch, and the polymers are synthesized by enzymatic addition of sugar molecule on pre-existing oligosaccharide chain through glycosidic bonds. The properties of polysaccharides generally depends on the length of the chain and the branching, presence of regular secondary or tertiary structure, whether the chain is straight or coil. Well, polysaccharide can be classified into three groups. First, basically they are classified into major two groups. One is homopolysaccharide and another is heteropolysaccharide. In homopolysaccharide, single kind of monosaccharide are present which are linked with each other by O-glycosidic bonds. For example, starch, glycogen, cellulose, dextrins all are composed of sugar molecule, but the linkages are different in all the examples. In starch and glycogen, both sugar molecules are linked with each other by alpha 16 and alpha 14 linkage, but the difference in between two are the number of branching. Whereas in cellulose and dextrins, branching are different from the glycogen and starch. In heteropolysaccharides, they may be of two types. One is glycosaminoglycans and mucilases. Again, the glycosaminoglycans, they are further classified into hyaluronic acid, heparin, chondroitin sulfate, serum mucoid, blood group polysaccharides, whereas in mucilases, agar, vegetable gums, pectins, hemicelluloses are kept. Well, students, now we will come to know about the functions of polysaccharides, how important they are in the living beings. They are very important because they play very important role in many ways. First, they provide shapes, structure to the cells, and they serve as protective covering of the cells. They also play important role in the form of energy. They can be stored for a long time in the form of starch, amylose, and they serve as nutrients in many ways. It may be their derivatives or it may be a free sugar. And they also play a very important role in cell-cell interactions and recognition. They can recognize self and non-self on the presence of the monosaccharides or oligosaccharides on the surface of the cells. So because they are very important, that's why they are widely distributed in all living beings, from the simplest bacteria to the complex animals. For example, in microbes, they are present in the form of xanthan, gallon, and dextrin. And in seaweeds, many kind of polysaccharides like agar, alginate, carrageenan, which we are using in daily microbial work and in food also. And in plants, they are present in the form of gum arabic, guar gum, pectins, cellulose, starch, and consic, etc. And they are present in the form of chitin, DNA, RNA, ATP, NADP in animals. Well, let's see some example of homopolysaccharides, oligosaccharides and polysaccharides. First, this is the example of homopolysaccharides. First example is starch that is generally present in plants in the form of food. It exists into two forms, amylose, in which glucose molecules are linked by alpha-1,4 glycosidic linkase, and in amylopectin, glucose molecules are linked by alpha-1,4 glycosidic linkase and with the alpha-1,6 branching. And in cellulose, it is a polymer of glucose linked by alpha-1,4 cellulose, it is a polymer of D-glucose linked by beta-1,4 linkase and serve as structural framework in cell walls of the plants. There is no branching present in it and possesses crystallinity in it. Whereas glycogen, it is again a branched molecule and it is basically amylopectin, a polymer of glucose linked with alpha-1,4 linkase and with a branching at alpha 1,6 linkage like starch, but branching is quite higher than starch. Therefore, it is much more soluble than starch. Due to higher branching, it is readily mobilized in immediate demand of energy. That's why in animal, glucose is stored in the form of glycogen, whereas in plants it is stored in the form of starch. Well, following are the examples of heteropolysaccharides. First, glycosaminoglycans, GAGs. The most abundant heteropolysaccharides in the biological systems are GAGs. They are polymer of disaccharides unit of N-acetyl galactosamine or N-acetyl glucosamine and the glucouronate or iduronate. GAGs are highly negatively charged and viscous, hence serve as good lubricating and shock absorbing fluid in the joints. Whereas pectins, they are present in fruits like apple, lemon and forms gel with sugar solid. They may consist of galacturonic acid, galactose, D-xylose, D-apiose and arabinose linked by alpha-1,4 glycosidic linkages. Whereas in hemicelluloses, they are mostly comprised of pentose sugars such as xylose, arabinose, ribose and few hexose sugars also. Hemicellulose are present in plant cell wall where it makes a bridge in between lignin layer and cellulose layer, providing rigidity and stability to the cell wall. Well, now we will come to learn about the physical properties. What are their physical and chemical properties? How do they look and how do they react? So, first thing, the polysaccharides and basically carbohydrates are readily soluble water. But if they are present in crystalline form like cellulose, hemicelluloses, agarose, which are very highly branched, they are readily soluble or insoluble in water. They are very viscous in nature, mostly used in the food industry as a thickening or gelating agent like agar and gums and resins. And they are very hygroscopic also. They contain about 2 to 10% of water even after drying. This property of alginic acid found in seaweed, it acts as anti-desiccant and makes it viable on hot beaches. And they are very stable except some reducing sugars. Those contain free aldehyde or hemiacetal group which can react and may be oxidized or reduced, which undergo oxidation and enolization during that process. And they are optically active because they contain multiple chiral centers. That's why they possess optical activity and are also present in enantiomeric forms like D and L glucose or distereomeric forms. And in chemical nature, they are very reactive. They may go in an oxidation process, they may go for a reduction process depending on the functional group present on that. So, in monomeric form, they may go for the acid hydrolysis. So, the glycosidic linkage, they are generally prone to acid hydrolysis and produces monosaccharides and their derivatives. Means if we react the polysaccharides or disaccharides with the acid, they may go for the acidic hydrolysis and the monosaccharides or their derivatives will be formed. They may be chelated with, in presence of metal ions. They react with the various metal ions like carboxylate, O-sulfur and O-phosphor may chelate metal ions. Alginate and carrageenan forms gel by cross-linking with the calcium ions and used for immobilization of enzymes. Copper chelates heparin and dermatan sulfates and affects their solubility. The free functional group such as amino group may undergo acylation, sulfonation or phosphorylation reactions and forms esters of sugars. In metabolic pathway, the glucose undergo phosphorylation process. Then the glycolysis takes place. Otherwise, without phosphorylation, the glucose may escape from the cells. So, phosphorylation helps to retain the glucose inside the cells so that we can get energy out of it. Let's know about the biological roles of carbohydrates. Carbohydrates play very important roles in various biological functions which can be categorized into followings. First, energy storage. Carbohydrates are simplest form of energy and provide instant energy from glycogen through glycolysis process, and it is stored in the form of starch in plants. Second is cellular recognition and interaction and signaling. Third, cell adhesion and extracellular matrix is a complex mixture of glycoprotein, glycoconjugates, glycosaminoglycans having multiple functions. All GAGs are covalently attached to core protein, thereby forming a proteoglycans PG with multiple functions like filling the space between cells, binding cells and tissue together, providing a lattice through cell can move, and they are very good source for filtrations. Means they allow only selected molecules to pass through them, like the cell when they are present in cell wall or in the junction of cell between two cells. And further function is structural framework, which is very important and which gives a support and elasticity also. They play important role in cell recognition, growth and regulation and differentiation also. The number of monosaccharides units are essential to distinguish between normal and abnormal cells in the tumor or in cancer cells. So they play important role in cell recognition and the signal transduction also. The molecule can transmit signal through oligosaccharides from one cell to another cell for the next step of the functioning or metabolism. As a structural function, many polysaccharides like celluloses, pectins, hemicelluloses present in plants and glucosaminoglycans, chitins in animals provide strength and structures to the cell and tissues. In this picture, the cell wall is composed of different layers of pectins, hemicellulose, lignin and cellulose. There is a bridge between cellulose and hemicellulase, and which is packed by lignin, and the protected covering of lignin is protecting it and giving a hardness to that structure. Whereas in bacteria, it is composed of, the cell wall is composed of N-acetylmuronic acid and N-acetyl glucosaminic acid, in which is present in an alternative network frame. They are linear, basically the polysaccharides which are playing important role in structure function are linear and also contains a highly ordered secondary structure. cellular recognition and interaction and signaling. Cell adhesion and extracellular matrix is a complex mixture of glycoprotein, glycoconjugates, glycosaminoglycans having multiple functions. All GAGs are covalently attached to core protein, thereby forming a proteoglycans PG with multiple functions, like filling the space between cells, binding cells and tissue together, providing a lattice through cell can move, and they are very good source for filtrations. Means they allow only selected molecules to pass through them, like the cell when they are present in cell wall or in the junction of cell between two cells. And further function is structural framework, which is very important and which gives a support and elasticity also. They play important role in cell recognition, growth and regulation and differentiation also. The number of monosaccharides units are essential to distinguish between normal and abnormal cells in the tumor or in cancer cells. So they play important role in cell recognition and the signal transduction also. The molecule can transmit signal through oligosaccharides from one cell to another cell for the next step of the functioning or metabolism. So students, let us conclude what we have learned till. So first we have come to know that carbohydrates are most abundant and diversified organic compounds found in living organisms. They play important role as energy stores, fuels, metabolic intermediates and cell recognition. Carbohydrates are synthesized by plants through photosynthesis using light energy and CO2. They may be present in mono, di, oligo or polymeric forms. Physical property depends on their monosaccharide, microsaccharide compositions, in glycosidic linkage, functional groups, molecular size and branching. And the modification in the structure of functional group leads marked changes in physical properties, whereas chemical properties respond accordingly to their monomers, functional group, linkage and configuration. Carbohydrates metabolism is the center of metabolic reactions. Hence, any disorder in their metabolism may lead to deadly diseases like galactosemia, glycogen storage diseases, GSDs, lactose intolerance, diabetes mellitus, etc.